a real pleasure to be here, and I appreciate the opportunity to share some new results with you. Let's see if I can, whoops, how do I advance this thing? Like that. Okay, I'm fascinated and somewhat obsessed with the biological process of recombination that happens during meiosis. To prepare for meiosis, cells replicate their chromosomes, and then they do this seemingly bizarre thing of creating self-inflicted damage in the form of many double-strand breaks, the minority of which gets repaired as crossovers among homologs. And there are many reasons to be interested in meiotic recombination. One is that in many species, it's required for proper chromosomal segregation. And a lot of the diseases that we see in humans are actually coming from issues with meiotic recombination. It shapes resemblance among relatives, and as this audience knows well, it influences evolution. There are pretty serious functional constraints on recombination, and yet the rate at which it occurs evolves. And this is just one depiction showing at a very high taxonomic level the log of the recombination rate on the y-axis there. And you can see there's a lot of variation in organs per megabase across major groups. That's also true when you look between closely related species, you see differences in recombination rate, the total number of crossovers in a meiosis, but also in the landscapes along the genome, as we've heard about in this meeting. And even within species, there's quite a bit of variation among individuals. In my lab, we spend a lot of time studying this question of how recombination rate evolves from an empirical perspective, and involves multivariate approaches that include the characterization of crossovers in individual gametes, the genetic mapping of recombination rate modifiers, both within and between species, and the molecular evolution of the recombination pathway. But I'm not going to talk about any of our empirical research today because we're at a math meeting. Okay, so one of the characteristics of the recombination pathway is that it involves the repair of double-strand breaks. Unfortunately, this repair is error-prone, much more so than repair of mutations induced during DNA replication, for example. And I don't want to get into the molecular details here, but I just want to say there's the generation of the double-strand break, there's a strand invasion followed by DNA synthesis, and double-strand breaks can get repaired as crossovers or non-crossovers. And it's, as I said before, a minority of them that are repaired as crossovers. But studies in yeast going back several years have demonstrated that this process is mutagenic. So there's a considerable elevation in the mutation rate associated with recombination. We can also see this in humans where we can use giant pedigrees to identify crossovers, identify de novo mutations, and look to see if they're correlated. And it turns out that they are. This is a plot from studies in the Iceland pedigree. The sex-specific mutation rate is on the y-axis, and the distance from crossovers is on the x, and they're separated into mutations near maternal crossovers versus those near paternal crossovers. And the dotted lines show the average genome-wide rate. And so what you can see is that there's a considerable elevation of the mutation rate near crossovers because of the processes I just outlined. And that falls off as you move away in distance along the genome from the crossovers. So this made us wonder, could the cost of elevated mutation overcome the advantage of increased recombination? We expect, based on what we know from the molecular biology and the studies of pedigrees in humans, that the mutation rate is elevated near crossovers, but that elevation has not, to my knowledge, been taken into account in models for the evolution of recombination rate, even though there's a whole separate branch of modeling that studies the evolution of the mutation rate. So to answer this question, we revisit and build upon a very rich tradition in population genetics of modifier models, and started as simple as possible with a deterministic haploid 3-locus model that has this structure. 2 loci affect viability, and those are the targets of selection. And then there's a modifier locus that is itself neutral, but is controlling rates of recombination and mutation at the viability loci. So it looks something like this. We can start with the idea that the resident allele, the uppercase M, is fixed at the modifier locus, big M. And in that background, at the viability loci, we have mutation from uppercase to lowercase alleles at rate U, and it's just in one direction, recurrent mutation to deleterious alleles. And then also in that resident allele background, we have recombination occurring at rate R1 between the viability loci and at rate big R with the modifier. This is just when the resident allele is fixed. And then when we introduce the modifier allele, little m, it modifies mutation and recombination rate. So this is the twist here. At the bottom here, you can see the recombination. Now we're going to go from R1 to R2 or R3. I'm going to sort of ignore this R3 part because it doesn't actually affect the dynamics. But so we're talking about transitions between R1 and R2. That's the evolution of recombination we're interested in. And the modifier allele changes the mutation rate by this factor D, little d. And then the next stage of the life cycle is selection. And selection, as I said, is just focused on the 2 viability loci with 1 minus S, 1 minus S, and then that double mutant class is the one where we get some interesting stuff by introducing epsilon for epistasis. And we're particularly interested, although the model is general, in this zone where we have negative epistasis, which has been shown to favor increased recombination over time. Okay, so the goal here is to conduct an evolutionary invasion analysis. We have 8 recursion equations. The first group of them specifies the frequency in the next generation of each of the haplotypes that contain the resident allele as a function of the frequency in the current generation, which includes mutation, recombination, and selection. And then what we're going to do is find the equilibrium for this case with the resident allele fixed, and then introduce the modifier allele and determine under what conditions it will spread. Okay, so if we just start with the resident allele, we can find the mutation, recombination, selection equilibrium, assuming epistasis is weak, and this has been done before. I'll just point out that the frequency of deleterious alleles, you have this first term here, which if the mutation rate is small, reduces to the usual familiar U over S. And then you've got this term that depends on epistasis. So what we're doing here is simplifying the 4 recursion equations for the haplotypes down to 2 variables, frequency of deleterious alleles, and also the linkage disequilibrium, which is the covariance of alleles at the 2 viability loci. Okay, so this is our equilibrium, assuming that epistasis is weak. And then the next step is to ask whether it's stable to the invasion or introduction of the modifier allele that changes recombination and mutation. And to do that, we write down the Jacobian matrix, which is now focused on those 4 equations that include the modifier allele. And we're going to evaluate this matrix at the equilibrium we just found. And this audience knows very well that if the leading eigenvalue is greater than 1, the modifier allele should invade. If the leading eigenvalue is less than 1, then the resident equilibrium is stable. So each of these equations is kind of ugly. And so a 4x4 matrix of ugly equations, is going to give you a really ugly leading eigenvalue if you can get to it. So to deal with this problem, we conducted a perturbation analysis to approximate the leading eigenvalue. And to do that, we assumed epistasis is weak and also the effect of the modifier allele is small on the order of epistasis. If epistasis is absent and the modifier allele has no effect, the leading eigenvalue should be 1. And so this logic suggests that we can approximate the leading eigenvalue as 1 plus some terms, where the terms measure the consequence of a small change in epistasis and the effect of the modifier allele. And then we can consider the terms to determine stability. Okay, so here's the term that involves epistasis. This is the piece where recombination is the dynamics of the evolution of recombination are important. And I just want to make one comment about this. If you just kind of look at the beginning of the equation, you can see, first of all, the difference in recombination rate is here, which makes sense, and that's weighted by epistasis. And as has been seen in previous modifier models, we see that this term is positive when increased recombination is favored, that is when epistasis is small and negative. So we recover that well-known result. But I also just want to point out that this term is proportional to epsilon times SU squared. Okay, so keep that in mind in terms of order of magnitude. Now, if we look at the term involving mutation effect of modifier allele, it's a bit simpler. And here, this term is going to be negative when the modifier allele increases mutation. That is when little d is positive. And you might notice that in contrast to the epistatic term, it's proportional to d times SU, linear in terms SU. So this term is bigger, to use a mathematical phrase. And what this means, at least in this weak epistasis zone where we expect recombination to be favored, is that the fate of the modifier allele tends to be dominated by its effect on mutation. So we probably shouldn't ignore mutagenic recombination when we think about the evolution of this trait. All right. How much time do I have?
You have 4 minutes.
Great, thank you. Okay, I just want to, mention another fun thing that we can do here if we are interested in how this might scale with the number of loci, right? This is a 3-locus model, but we can build a simple 2-locus model for that has a modifier of the mutation rate and a recombination rate between the modifier and one viability locus, and just compare these to see if there's anything interesting. And if we make lots of things small, weak epistasis, weak selection, very weak mutation, and make the recombination rate equidistant between loci, then what we get for that mutation effect term for the 3-locus model looks like this. It's a big simplification from before. And we can do the same thing with the 2-locus model, we get the same term. And so this is just fun because it suggests that as we add loci, that we're getting some real sort of intuition about how the effect of mutation should scale in terms of whether the modifier will invade. So now I'm just going to show you in the last few minutes a few results from numerical analyses. And so these are exact and don't make any assumptions about the strength of epistasis. And what you can see on the y-axis here is plotting the leading eigenvalue -1. So when you're above the horizontal line, That's invasion. When you're below, it's the equilibrium stable. This is the case that's already been solved where D equals 0, no mutation effect of the modifier. And here I'm looking at tight linkage of the modifier, 20% increase in recombination. And if you're interested in the details, you can look over here. I'm just going to sort of focus on this space. So if we introduce a small increase in mutation rate, it begins to narrow the advantage of increased recombination already. But as we introduce what I think are more realistic increases in mutation, we pretty quickly overcome the advantage of increased recombination entirely. Even if we look at a big increase in recombination, really big, which should be favored even more, it's inhibited by elevated mutation. So the red versus the blue lines. And when recombination is low, this is, the advantage of the increase in recombination is stronger, but in that empirical space, the effect of the modifier on mutation is expected to be bigger because you're closer to the crossovers. And so even though you see this increase in invasion probability, because in the situation where recombination rate is low, it really gets diminished by what we would expect from the effects of mutation. And even in the zone, well, things get even worse when you consider that a situation where the modifier is loosely linked. This is something that's already known. It's hard to get the spread of a modifier increasing the recombination rate unless the modifier is closely linked. If we relax that assumption, it's even harder to evolve increased recombination rate. Okay, so hopefully the implications are clear, but I just want to spell them out. Error-prone DNA repair of double-strand breaks can inhibit the evolution of increased recombination. Models that do not require epistasis, of course, might allow the evolution of increased recombination under broader conditions. We know that from a lot of theory, but we suspect that the mutagenic consequences of recombination will generally inhibit its evolution. And recombination increasing alleles that spread, I would predict, might be concentrated in parts of the pathway where the pleiotropic effects of mutation are lower. And just to finish up, I'll show you that there's a sign, I think, that this might be true. If we look at genome-wide association studies from humans and other mammals, variants associated with recombination within populations tend to cluster in genes that do not change double-strand break number, right? One way to increase recombination rate is to change the number of double-strand breaks, but then your little d is going to be big. So this is already sort of consistent with what I've told you. We can also see signs of this molecular evolution signatures, like so nicely explained in the last talk. Amy Dapper did this work in my lab where she looked at the molecular evolution of the recombination pathway across 16 species of mammals and broke them down by step. So moving from left to right, roughly speaking, things get less mutagenic. And one thing that she found was that even though there are rapidly evolving genes, these red ones in different parts of the pathway, there's a real enrichment of them later on, synapsis, crossover, non-crossover decisions that are suspected to be less mutagenic in the pathway. So with that, I'd just like to thank Sally Otto in particular, who has been a crucial source of inspiration and mentoring, learning how, teaching an empirical geneticist how to build mathematical models. So I feel greatly indebted to her. I'm indebted to University of Wisconsin, which is a wonderful public university, sources of funding. And I also just want to finish by saying how much I appreciate all of Joanna's efforts, the organizing committee, Jen, for a truly outstanding and provocative meeting, and to Noah Rosenberg in particular for really leading the founding of this wonderful society, which I think is going to have a very bright future. Thank you.
So I know this way of modeling recombination is traditional. But I got very confused when I learned some cell biology, given that most organisms have one crossover per chromosome arm or one per chromosome. So the human map distance, I can't remember exactly, but it's 46.001 or 01 or something, right? So that there's variation seems to be very limited insofar as there is variation. The biggest evolution in the rate of recombination is in the number of chromosomes. So that's yeah, and then related to that is also, you know, so I guess what I'm trying to say then is if you are locally changing the recombination rate to create or inhibit, you're simultaneously changing, you're not changing the genome wide you're usually changing whether it happens here versus somewhere else. On that point, recombination usually occurs in vertebrates near genes. And I know Lawrence Hurst has proposed that's because GC biased gene conversion is advantageous. Otherwise, we'd just be too AT swamped in the coding regions.
So I would those are good points. I would just say that the fact, you know, in vertebrates that recombination is occurring closer to genes should make this problem even worse.
Yeah.
Right. But your point about cis, I should have spelled out that this is a modifier model. You know, you're looking at local pieces of the chromosome, and we don't know how you get from a whole bunch of cis modifiers to, you know, the full picture. There are trans modifiers that this is completely ignoring. So that's right. On your first question, I'm not sure. I think definitely there are big changes in the effective amount of recombination with variation in chromosome number, but there are also big changes in recombination rate, even keeping chromosome number constant. So, and that's part of what we're trying to explain here.